to our next speaker, Dr. Ajay Kohli from IRRI, and he's going to take us from maize now to rice. His title is a long one. I won't attempt to read it, but I can see that he's not ready yet. Elaborating a unique reglon for rice yield under drought. Components of genes underpinning or affected by transcriptional, post-translational, biochemical, and epigenetic mechanisms.
Morning, everybody. Thank you very much for this opportunity. And coming from a family of classical Indian musicians, I want to take this opportunity of congratulating the organizers for a fantastic cultural evening yesterday. It was a wonderful evening. With this kind of a title, I hope I'm not the only one that will cross the limit for 10 minutes. Quickly moving on, we are supposed not to talk a lot about what everybody knows here, but I just want to quickly mention that the genes that we identify for drought tolerance are not always part of the QTLs that we know, are not always part of the genotypes we know, are not always part of the organs where they function, and the morphophysiology mechanisms are not the same, the soils are not the same. So it's a very complex issue that we are dealing with. And as a molecular biologist, I'm just concentrating right at the end in the genes, where again, if we look at the drought literature, the genes that have been identified as possible sources of drought tolerance belong to all different categories, all different annotations, metabolic pathways, different aspects. One way of reducing the complexity was approached by Gary Atlin's group in the beginning when he started at IRRI, and that was to sort of look only for yield under drought. And it has had a massive success. One of the QTLs that was identified has been used in the field now, and it has been taken forward after Gary left. But the QTL is actually valid in multiple genotypes, multiple organs, multiple environments. And the point I want to make is that something like that could not be a single gene QTL. So just to quickly sort of mention the names, Jerome Bernier, who was the graduate student of Gary Atlin, was instrumental in identifying that. These people, Gary himself, Venu, and Arvind, Arvind has taken this whole work forward, and Amelia's group has done a lot of morphophysiology studies on that. What we could show that if we have different genes at the QTL, which we know because of deep sequencing of the region, actually, we could get all the polymorphic genes, or most of the genes were polymorphic, along with the markers that were used. What we can clearly see, if you go down bottom, at the bottom between, these are the recombinant lines under the well-watered and the drought condition. The higher yielding lines are the ones where you have the entire QTL present. When you map that, some of the genes come up to be very useful, and the highest score is for the no apical meristem transcription factor. But what makes this no apical meristem transcription factor very interesting is the fact that there are at least 6 other genes, the targets of this transcription factor, sitting right there at that locus. And so we use that to do the transgenic analysis, and we could show that the transgenic plants would actually recap the phenotype of the nil that was made with the QTL, mainly for increased panicle branching, increased root branching, better water use efficiency, but not similar levels of yield, which again says that it is a multi-gene QTL. One of the genes that we found interesting, I won't go through describing the whole thing, is a gene called the glycerophosphodiester phosphodiesterase. And we have initial results to actually show that it is very highly expressed in the floret regions, and within the florets it is expressed in all the parts of the florets, anther, ovule, stigma, lodicule, filament. So this is where the biochemical aspect of it comes in, where I say the biochemistry is involved. What it really does, we are not really very sure, but enzymatically it actually converts a certain compound of glycerophosphodiesterase into 3 glycerol 3-phosphate and in the corresponding alcohols. And it is generally upregulated in the P deficient conditions. It's very similar to the kind of a phospholipase interaction, but we don't know the details of how it might be interacting. However, having it expressed in the reproductive organs makes a big difference. The next bit is about the transcriptional aspects. Of course, the neoapical meristem transcription factor by itself, by its nature, can bring about a lot of changes in the transcription of the genes that it regulates. But there is a serine threonine protein kinase, which is also a target of the same transcription factor. And when we look into more details of that, it can actually phosphorylate and activate the SR proteins of the spliceosome. And we know now there is enough literature to suggest that under drought, there is a lot of alternative splicing going on. And the diagram actually just shows that it is autophosphorylated. It can phosphorylate the SR proteins, which we have isolated and done in vitro assays. And it can also phosphorylate the no apical meristem transcription factor. Although this is in vitro, we are doing phosphoproteomics to figure out what are the other targets of this kinase. The next one that I'm going to talk about is related to the epigenetics aspect. I will come back to why we thought it was related to epigenetics, but we have actually now clearly shown that this amido hydrolase is actually the first plant guanine deaminase. The guanine deaminase activity is known in plants in like tea and coffee, where it is a precursor for xanthine synthesis. Its activity is a precursor for xanthine synthesis. But the gene coding for guanine deaminase was never known, and this is the first time we have been able to show that. This here is where guanine deaminase acts, converting guanine into xanthine, but it is connected upstream to a pathway which can affect DNA methylation. The reason we got here was because this protein is very similar to actually this enzyme, this enzyme, which can convert adenosyl homocysteine into inosine homocysteine and ammonia. And SAH is a potent inhibitor of DNA methyltransferases. So initially, our hypothesis was that this is what the amido hydrolase is doing and affecting epigenetic aspects. It did not do that, and we could finally able to characterize it as a guanine deaminase, and yet it is a downstream pathway, a downstream enzyme in the same pathway, and we have been able to show, among many other procedures, that the methylation is actually affected. I won't go in describing the details of the figure, but methylation is affected in the knockout plants. What is also interesting is both the serine threonine protein kinase and the amido hydrolase affect the lateral roots and the root hair concentrations under drought. And the last bit I want to talk about is the post-translational modification. The NAM itself is actually post-translationally modified. And again, without going into the details of what you're looking at, all I want to quickly show is that when the NAM is identified through monoclonal antibodies, you see a difference in the kind of spots that you get on a 2D gel when it is treated with a sumo protease or with an alkaline phosphatase, actually showing that the transcription factor is sumoylated and it is phosphorylated, and there is enough literature to suggest that these two activities are interconnected. It is the most important aspect of that is that it is differentially sumoylated under drought. Again, if you will remember, these are the spots which are actually changing, but also you can see there is new spots coming up in the nil lines which are not there in the donor variety. So there is a lot of post-translational modification also going on. This is nearly 8 years of work. I'm sorry if it is a bit confusing. I've gone through a lot of stuff very quickly. But essentially what I've tried to say is that there is a region which has all these genes sitting there, 4 out of which are responsible for different mechanisms which are very intricately involved in drought tolerance, and we know that from literature. Transgenic validation has been done for NAM, of course. We have knockout lines for some of them. We are making other transgenic plants. For those which we have activation lines, we are making knockout lines. For those which we have knockout lines, we are making overexpression lines. We have done omics data also on the nils, on the transgenic plants expressing the NAM, and comparing it to the parents, of course. We've got data from root, leaf, panicle tissues. Some of this data was presented last inter-drought. One of my postdocs presented the initial data on that. And I would like to just quickly wrap up those results by saying some of the things which might raise some discussion, that osmolytes, which we are very fond of in terms of drought tolerance, proline, trehalose, other raffinose, sugars, etc., they do not accumulate when you get yield under drought. Genes are differentially expressed in all the different tissues. The same gene is differentially expressed in different tissues. We had a lot of evidence of that. Panicles, the panicles do not change much if you are getting the yield. And in our case, with this QTL, we are getting the yield as good as in the parent. They do not change much. I will not mention a lot about the other two. The protein activity is a layer of differentiation. We know that from our studies on amino hydrolase. So this is just a slide I will leave you with. And I quickly mention some of the people who have done the work and our collaborators. Thank you very much.